continue with identification of snakes with highly toxic venoms through nucleic acid testing of toxin genes, bioinformatics study to design primers for isothermal PCR of Bungarus candidus toxin gene. Presented by Mr. Antonius Reuben.
Okay. Hello. Good afternoon, the honorable judges, the fellow participant of the conference. My name is Antonius Reuben, and I am a medical student from Indonesian Defense University. And for the next several minutes, I will be talking about my team study of one of the common inhabitants of the forest and the mountains, which are snakes. Why do we choose snakes on our studies? What is the significance? What's the special of these snakes? And my answer towards it is one word, and it is called wandering. Why? Because I believe we all agree that all great technologies, all great innovation, and all great theories start with an act of wandering. I wonder why an apple falls from trees onto the ground. I wonder how we can make travel less time consuming. And for me, in these studies, I wonder how those TV shows and YouTube channels can identify those snake species by their appearance, such as color, patterns, how they move, how they act, how they strike their prey. And this makes a challenge for my team to learn and study about the identification of snakes. And while we are searching about the snakes, we found a study that mapped the distribution of the venomous snakes on a global scale. We can see the red color indicating a high number of the red color indicating a high number of venomous snake species, and green indicating a much lower venomous snake species. While we were studying about this graph and this picture, we realized that these maps concentrate on regions such as Asia, and we also realized that there is a low to minimum effort by scientists on learning and studying about venomous studies in regions such as Asia. This makes us feel shocked because let us think for a minute. While these people live in this vulnerable state, and why there isn't any scientists or researchers to studies about this situation. And this makes us also realize that we as an academic community must raise this threatening situation, and we must raise an awareness towards the senior and fellow researchers and scientists for realizing the situation and also partaking into studies about snakes in the Asia region. So back to the topic of identification snake bites. How do we make it possible? As we all know, snake bites flows through the bloodstream in quite quickly, and we need a rapid assay towards diagnosing what snakes have bitten this patient. So while my team are searching about how to make an assay as rapidly and as efficiently, we found out that there is a method called loop associated mediated isothermal amplification, which are already being accepted and being used for several studies such as HIV, Ebola, tuberculosis, and etc. So we wanted to make this into a reality, so we use it on making this isothermal amplification on diagnosing the snake. So the first step is we making the primer design by gaining the gene sequence through gene bank, and we use it on an obelem primer designing tool, and after it we test it for the specificity so that it can be used for diagnosing and rapidly for the early treatment. And after it, this method can also be used to rapid diagnostic for other snake species, such as maybe viper or cobra. And the way to do it is we just change the gene sequence, and we will make the primer that are also able to diagnose the snake bites. By doing this, we can help and supporting development on applicable method towards efficient analysis and diagnostic to reduce mortality by misdiagnosis. Thank you. That's all for my presentation. Thank you for your attention. My name is Antonius Reuben, and have a good afternoon.
One of the finest. Any question from our judges?
So you use the venom from the snakes to test your kits, right? The venom is from the snake.
Yes.
Yes. But in the real situation, the snake comes and bites the patient and goes very quickly.
Excuse me, can you elaborate
But
much
in
further?
the real situation, snakes comes and bites and escape very quickly. How can you catch the snakes in the real situation? So have you tried to test the from patients?
Oh.
Yes.
Yes, this method is still on developing. This is the first step towards making it into a reality, which are the primer designing, and later we will make the in vitro testing to make it if this primer is really and suitable for this gene of this specific snakes.
What is the standard method for detect the snake toxin?
Excuse me, I didn't quite get it.
What is the standard method for detect the snake toxin? I mean, normally the snake toxin can detect by with standard method. Use molecular method or protein detection.
Okay. So in these studies, the snake toxin will be detected by the primer, and we will make a control positive. So if it detected, if the result shows the same color with the control positive, and so it's shown that this guy, that this person has been bitten by this particular snake species.
And if you want to develop this method, what you want to compare with the standard method?
This method will be compared with the PCR and use and other DNA diagnostic method. Thank you.
Thank you.
Thank you, Mr. Reuben.